So first of all, it's been an absolute pleasure to have you here, and hopefully we are going to have you again in November and December and for the many months to come. And I think, Ravi, you've been able to secure seminar speakers, for the next, I think, 5 or 6 months. So there's going to be a lot of activities related to the PHRN, and we've been trying as hard as we could to identify a date and a time for having a PHRN Research Day. If some of you may recall, last year in November, I believe it was last week of November, we had the PHRN Research Day. We've been trying to hold it also in the last week of November, but I think it probably would overlap with Poultry Industry Council Innovations Day. So as a result of that, we are thinking that perhaps it would be a good idea to work around that day and maybe even reschedule it for sometime in January. But just wanted to give you a heads up that there's going to be some announcement coming to your email shortly, so please don't delete it. And I think I also owe you an apology because it seems that your email system has been bombarded with emails from various different sources, either from Ravi, from myself, or from OVC Communications. And on top of that, LRIC and PIC, they've been bombarding you with emails about today, and hopefully we are going to be able to streamline. But that's actually a good segue to the next part of my talk. And I have to make a disclaimer here. I'm not a communications person, nor do I have any sort of formal training in communications, but what I have actually come to a conclusion is the fact that we require lots and lots of communications. Not communications among researchers, because I think we, by and large, do a really good job of communications with our colleagues and peers via, for example, publishing papers or via attending conferences, advisory committees, and so forth. But we have done, with all due respect to my colleagues and myself, I think we've done a job that can actually be improved upon in terms of communicating with our stakeholders, be it private industry, be it the public, or be it the government. I think, by and large, we have lots of things to offer, and we can definitely improve upon that. And as Ravi alluded to, one of the main objectives of the PHRN is really to streamline pipeline of communication and be able to communicate with various different layers of our stakeholders in the research community, in the academic community at large, to the public, to our producers, and also to the government as a whole. So as a result of that, this is basically just to give you an idea of our communication pipelines as it stands at the moment. Ravi was recruited 2 years ago, less than a year ago. I'm actually jumping far ahead of myself now. So he was actually, don't get scared. So Ravi was actually recruited less than a year ago, and some of you who came for his presentation, I did actually give an overview of where he came from. He got his PhD from Guelph with Dr. John Prescott, went on to do a postdoc partly with me and also eventually at mRNA Vaccine Center in Atlanta. So we've been very fortunate to have him back in Guelph. And Ravi has been very active on a wide variety of different activities related to PHRN communication pipelines. Many of you have already seen our website. Our website has gone through a huge amount of revamping with the help of Natalie Lumio in our department and Ravi, who have basically turned this around and made it quite professional. And this is actually an area that I'm playing with members of the PHRN to help us strengthen and fortify, because I think this is probably going to be one of our main faces with the public, even with our own peers and colleagues. So we do actually want to make this as professional as possible and as comprehensive as possible. Ravi has also been very active tweeting things. If he had asked me a few years ago, do I know anything about tweeting? I would have said no. Do I value tweeting? I would have said absolutely not, because it has no scientific value. But in actual fact, the public actually sees all these things, and they take it with and they take it at face value, and they do actually appreciate, and they do actually place a lot of value on what we are able to communicate with them. So Ravi has also been very successful in hitting various different poultry magazines and trying to insert some of our stories, some of our success stories, because I think, by and large, I'm sure that you would agree with me that we've done a really good job of, like I said, publishing our papers. Each one of us has probably an H index of whatever, and we've published whatever number of papers in scientifically reviewed journals. But in terms of how well have we actually been able to penetrate some of the poultry magazines or commodity magazines, I think that remains to be seen. But I think in my own case, I have not actually been able to do a good job of communicating with our with the public at large or with producers. And that's in fact been an area that Ravi has been working on, to share success stories with various different vehicles for communication with our producer groups. Other social media outlets, and this is also something that Ravi, as well as Karen Mantell, who's the communications officer for the Ontario Vet College, has been helping us with, is to penetrate into social media outlets. And there are many of these that are available to poultry specialists, to poultry researchers, or to producers for that matter. And we've been trying to highlight and showcase some of our success in those social media outlets. And internally and also kind of externally, so I do consider PIC and ELRIC sort of actually integral to our success, and I call them internal sources, but at the same time they're not actually affiliated with the university, even though we do actually want to embrace them as much as we could. PIC and ELRIC has also been very helpful to us in terms of sending our success stories to the community at large. OVC has in fact committed substantial amounts of funding, as far as I'm concerned, a substantial amount of in-kind contributions by providing the help of one of our communications officers, Karen Mantell, and also the other communications officer who, whenever possible, she does actually contribute to the PHRN and success of PHRN communications. And I'm also pleased to tell you that now we have some interface with the Office of Research. So Office of Research is now coming to us for stories in relation to poultry, and we are more than happy to share any stories that you can share with us, with the Office of Research, and like I said, the community at large, the scientific community or otherwise. This is just a new look of the PHRN website, and like I said, this is a moving target in the sense that we do actually want to update it on a daily basis, or maybe not daily basis, but at least weekly basis. So please do contribute to the website. If you don't like anything in there, please let us know. If you like something about it, please let us know. Please tell us what we can do in order to strengthen the website and make it look better, because like I said, believe it or not, this is actually our interface with the public, and this is how they're going to be looking at us. And Ravi has been very, very successful, I have to say. So before he came along, I had created a Twitter account, and despite my lack of enthusiasm for tweeting, I started tweeting, and I said, oh God, what am I doing with this? But Ravi came along and he said, we've got to make sure that we have enough followers, because if you have enough followers, then you're going to be able to have more penetration into the public. As a result of that, we raised the number of our followers from, I believe it was 50 or 60. So that was actually all attributed to my hard work. But during the last few months of Ravi's hard work, he's actually been able to take the number of followers to one order of magnitude higher. So he's actually been able to increase the number of followers from 70, 60, 70, I can't recall what it was, to over 700. And this was as of the weekend. And the number actually grows quite steadily. It has actually been growing quite steadily over the last few months or so. And this is also another source for communicating with our public, with the general public, or with our own colleagues and peers. So if there are anything at all that you would see interesting for the public or for the science community at large, then please tell us. I think we've been doing this rather actively and sometimes passively, but I think it would be a very good idea to have a combination, to have some sort of a hybrid structure. So Ravi, in some instances, he's been actually going after people and going after their papers to see what papers have just recently been published. But and I know that by the time a paper is published, it's already old, because it's probably been in the process for maybe a year, maybe more than a year, give or take. So we do actually want to make sure that we can communicate with our audience as swiftly as possible and as appropriately as possible. And I think tweeting is one way of doing that. And in terms of other social media outlets, I'm actually quite pleased to tell you that now we have connections with OVC Twitter account. So they do our tweeting and we do their tweeting, and at the same time, Office of Research is also connected to OVC Twitter. So everything that we do is actually going to be reflected, part of it, not all of it, part of it is going to be reflected on the OVC Twitter account, and the OVC Twitter account is actually connected here. So just to give you an idea, this is basically, I think it was, again, over the weekend that I downloaded this, that I printed the screen here. So the Twitter account on the Office of Research website, this is the main page for the Office of Research. It had some information about story about avian influenza. It also had some story about the Poultry Health Research Network. So I just wanted, this was only an example, just wanted to tell you that we can actually do something like this for any of the stories that you're able to share with us, and we do actually invite you to do that in the future, in the near future, and some of you have already done that. Mix is one of the social media outlets that we've been trying to penetrate into, and we are now subscribed to Mix. Mix is subscribed by, I think, over a few thousand people who are interested in poultry. And it sends out emails and notices and announcements and things like that in relation to anything poultry production management, welfare, health, disease, et cetera, and people can communicate their results, and they can also comment on announcements that are made or research discoveries that have been made and communicated. So this is actually one of the ways that we've tried to, like I said, communicate our success stories with the public. And there are multiple of these, and I'm not going to go into a lot of details, and some of you might have already seen our success stories published in poultry magazines and so forth. But this is another outlet that I thought I should tell you about. And many of you got the invitation, I hope, for the PHRN barbecue. It was just, I guess, it wasn't a random event that we invited the president, but I felt that probably the president would not accept our invitation, and lo and behold, he did actually accept the invitation. So the barbecue event was a joint effort between PIC, ELRIC, and the PHRN, and we invited Dr. Vaccarino, our new, well, no longer new, but almost brand new president, and also we invited Dr. Malcolm Campbell, who's our VP Research. This gentleman over here, and this is Dr. Vaccarino. We also had in attendance Ed Verkley, who's the chair of PIC at the moment, and Keith Robbins, as well as many other people from industry and from academia, and also to some extent government. Dr. Carrie Lissamore, who at the time was the interim dean of OVC, also gave a talk to welcome people, and I was actually quite pleased to see our president and vice president of research at an event. And I can tell you that over the last 15 years I've been in this university, I've never actually seen both of them at the same research event. They might have actually been, at various different other kinds of events, but not at a research-oriented event. And I felt that, this basically attests to the commitment of the university to our partnership with industry, with PIC and with ELRIC. So I just wanted to give you, I guess, a piece of good news that, we are definitely on the radar screen of these two gentlemen who are on top of the research enterprise of our university. In terms of our reach, I'm absolutely delighted to tell you that you have been able to reach out to almost every part of the globe. This is essentially, all these balloons represent collaborations between our colleagues, within our members, within the PHRN network, and various different continents and countries. And depending on the intensity of these balloons, you can appreciate the intensity of our collaboration. So we are basically in all continents, probably except the South Pole. Is South Pole a continent? I think it was actually a continent. So here we go. We do actually have our reach in all various different continents. And if you ask me what the next step is going to have to look like, especially in terms of expansion of our research and education, I would actually argue that probably we are going to have to maybe put some focus on this area, which is Middle East and Far East, and also definitely put some emphasis on South America, because these have actually been underserviced by our members. And if we do actually have any international aspirations, then my argument is that you're going to have to perhaps put more emphasis on this part and this part, because there is quite a bit of activity going on in terms of poultry production and poultry research to some extent. So that's basically all I wanted to tell you about the poultry network. And I know that I can't really shut my mouth at this moment. I'm going to have to talk about poultry research and what we have done in my lab. So what I wanted to do now is to talk to you a little bit about some of the work that we've done in my lab, and just to give you an overview of poultry research in my lab. And I don't actually do anything other than poultry research. And I have to say, at some point, I betrayed the field of poultry research, and I did maybe 2 years or 2.5 years of work on the mouse model when Ravi was in my lab, but that was actually a thing of the past. Now I'm 150% concentrated on poultry research, and this is just to give you an overview of my program, or the programs in the lab over the last little while. So there are, I would say, two major areas or two major programs in my lab. One is viral immunology, and the second one is gut health. So within viral immunology, we look at two viruses, Mareks disease virus and avian influenza virus. And I see that Grant is sitting here, so I'm going to say influenza A virus, Grant, that infects chickens. And we also work on gut health, and specifically we are working on development of probiotics. I'm actually quite pleased to see Jennifer Brisbane sitting here. She was actually a technician by the time she joined my lab. I believe it was 2003, then continued to be a PhD student, defended her PhD, and then I couldn't really let her go. So she was a postdoc in my lab, and she was critically involved in development of various different aspects of probiotics and gut health in my lab. The second part is a more emerging part of my program, which has to do with vaccines against foodborne pathogens, and specifically I'm talking about Campylobacter. We don't really have an active program on other foodborne pathogens. This is the only foodborne pathogen that we are at the moment concentrated on in terms of producing vaccines against it. So just to give you an overview of the various different programs that I have in the lab, that we have in the lab, it's actually my students, so I have to give them credit for that, not myself. Mareks disease virus is a very interesting virus, interesting from a wide variety of different aspects. It has some applications also in the field, because many of the chickens we eat, or many of the chickens we eat their egg, have already been vaccinated against Mareks disease virus, and there is the, not fear, but there is the concern that Mareks disease virus over time changes its virulence, and at some point of time it may become more virulent. And it has actually proven to become more virulent over the last 40, 50 years since the introduction of vaccines. Mareks disease virus is a virus that causes lymphoma in chickens, and the fact is this is actually the very first virus or oncogenic virus for which there was a vaccine available. So a few decades before Gardasil, which is against the vaccine against papillomavirus, there was a vaccine against Mareks disease virus, and it was a very efficacious vaccine at a time, almost 40 years ago, 40 to 50 years ago or so. And then eventually that vaccine became obsolete because it couldn't really render animals resistant to the disease. And then came along another vaccine, and then came along another vaccine, and now we have a few more of those vaccines, either in the pipeline, to some extent successful and to some extent not. And we now have many different strains of virus, or shall we say pathotypes of this virus, that each one of them has different pathogenicity, and some of them are quite virulent. So the work in my lab is really concentrated on two things. One of them is to have a basic understanding of how the virus behaves when it goes into various different tissues of chickens, how it behaves, because it actually goes into various different tissues and it behaves quite differently in those tissues. So what we want to know is how the immune system sees the virus, how it interacts with the virus, and how it responds to the virus. And when you think about MDV, it actually goes into various different tissues that I just mentioned. It goes through First of all, it's a respiratory contracted virus, so the virus has to go through the respiratory system. When it goes through the respiratory system, then it goes to lymphoid tissues. In lymphoid tissues, it activates some of the T cells, and at the same time, it kills some of the lymphoid cells. So as a result of that, the chickens that become infected are immunosuppressed due to the fact that some of their cells are going to die due to the presence of the virus. And then the virus undergoes the process of latency, meaning that the virus is there, is sitting inside the cell, but it doesn't do anything to the cell. But at some point of time, the virus comes out of latency. And when it comes out of latency, it can transform the cells, meaning that it causes neoplasia or cancer in those cells. And the cells that fall victim to the virus are T cells, or cells of the lymphoid system that become transformed. And once those cells become transformed, then they actually behave like any kind of lymphoma in humans. They go into various different places, from the eye to the brain to the tip of their toes. It can go to everywhere, those cells. And those cells can cause disease. And clinical signs and gross pathology of disease are associated with presence of those cells. But interestingly enough, the virus also goes to the feather follicle and to skin epithelium. And then this virus is actually not able to go through its productive cycle, meaning that the virus cannot be shed to the environment. And this is actually the place that a lot of vaccines can't actually work. So the vaccines can suppress the virus, and they won't allow the virus to come out of its latency. But the virus, no matter what, can go to feather follicles or to skin epithelium, and then it can shed from those tissues. And vaccines can't do anything about it. That's actually one of the reasons that vaccines have been successful to some extent to suppress disease and to reduce disease. But they have not been very successful in terms of reducing virus shedding into the environment. And when you have virus transmission or virus shedding, then potentially you have virus shedding. So one of the important things that I think we could do is to do things. There are two things actually that I think we can do. One is that we can prevent the chicken from being infected, and the other one is to prevent the virus from being shed from feather follicles. And that's actually been my holy grail. I've been actually really trying to get at this for a long time, and I hope that before my career ends, I've actually been able to at least find some answers to these two things, to be able to find a way to prevent the virus from infecting a chicken in the respiratory system, and also being able to somehow curtail replication of the virus and shedding of the virus from skin epithelium or from feather follicle. And just I tried to be a little bit high tech, and I had a video from one of my former students who's at the moment an associate professor at the University of Calgary. Let me see if I can. So the two chickens that you see here are control chickens, and my apologies for the high noise here, background noise. So the two chickens you see here are control chickens. And this is actually what you would normally find with a chicken that has been infected with MDV, and it shows the clinical signs of MDV, which is paralysis. And this is a typical type of paralysis you would see in chickens infected. One thing that is very important to note here is that this was also one of the first diseases for which we had breeding programs, or against which we had breeding programs. What I failed to tell you was that these two chickens both have actually been infected, but the chicken that is showing clinical signs of disease is of a particular genotype. And because of this particular genotype, this chicken is showing clinical signs of disease, whereas this other chicken has also been infected, but it has the so-called resistant genotype, and that's why the chicken is highly resistant and it's not showing clinical signs of disease. But both of them have actually been infected, and I can also tell you that both of them are not shedding the virus, irrespective of showing clinical signs. So what you would find in a vaccination situation, is that you would actually find chickens that look like this one, somewhat irrespective of their genotype. So what you do essentially is that you prevent them from showing clinical signs of disease, whereas both of them are infected, all of them are infected or could become infected, and if they become infected, they can all shed the virus. So the situation for vaccination is somewhat similar to what you see for genetic resistance to disease. So in the context of avian influenza virus, specifically we are looking at viruses of low pathogenicity because we don't have the capacity to work with highly pathogenic viruses. So we are specifically looking at these low pathogenicity viruses. These are viruses that usually infect waterfowl or infect chickens, turkeys, and so forth. And the viruses that we are working with are in fact from avian origin. So we do know that they're avian influenza viruses, and we put them in chickens, and lo and behold, they become infected, and then we can look at the rate of infection and determine whether or not they're highly infected or they have lower amount of infection. And there are two things that we are doing in my lab. There are two, let's say, arms of the study in my lab. One of them deals with developing standalone immunomodulators that can reduce virus replication, and the other one is to develop novel vaccines. I'm just going to tell you just a little bit about what I meant by these immunomodulators. I'm kind of actually preaching to the choir here because I'm sure that many of you are aware of toll-like receptors and microbial products that can activate the innate immune system. But just for the sake of conversation, I'm just going to briefly tell you what we are currently working on. So this is, if you can imagine, this is a bacterium, and if you start peeling off cell wall and cell membrane of that bacterium, you're going to see lots of different structures. There are also things attached to some bacteria, like for example flagella, which makes bacteria motile. You would also find RNA, you would also find DNA within those bacteria, all of which can actually stimulate the innate immune system. And here I'm talking about all species, almost all animal species. So within bacteria, you would find, as an example, DNA. Within that DNA, there are certain motifs which we call CpG motifs, and those CpG motifs, when they're unmethylated, they're highly immunostimulatory. Same thing actually holds true for things that you would find here in form of peptidoglycan or in form of lipopolysaccharide. Those are highly immunomodulatory, and flagella much the same way. So just to give you another overview of this, this is a gram-negative cell wall. And if you take a look at this sandwich of things, you would be able to find many different components, from the plasma membrane to peptidoglycan, and in the outer membrane you would find lipopolysaccharide, and somewhere in between you also find lipoproteins. And I can tell you that anything that I can't really say anything, but a lot of things that you would find in cell wall are in fact immunomodulatory or immunostimulatory. So for example, peptidoglycan is highly immunostimulatory. LPS or lipopolysaccharide is also highly immunostimulatory. These lipoproteins can also stimulate the immune response. So my lab over the last little while has utilized some of these molecules to determine whether or not by stimulating the innate immune system, we can in fact reduce shedding of the virus in those chickens that are treated prior to becoming infected with avian influenza virus. And the answer is yes. So this was actually the work that Neda has done, and I think, yeah, here she is. Neda has done in my lab, and in this work we've looked at some of the molecules that I just described to you, and some of them still have their commercial names and so forth. But just to give you an idea, this is lipopolysaccharide. This is CpG. So perhaps you remember just a few minutes ago I said within the double-stranded DNA of bacteria you would find CpG motifs. These are areas of DNA that are enriched for C and G nucleotides. And when they are unmethylated, they have the capacity to stimulate the immune system. So when you look at LPS or CpG compared to chickens that are treated with PBS, which are controlled but then became infected with AIV, low pathogenicity AIV, you do actually appreciate the fact that there is a reduction of approximately 2 logs or 2 log 10s, so approximately 100 times less AIV replication in their intestine or in their upper respiratory tract. So by and large, we've proven that by using some of these ligands or by using some of these microbial products that are basically available in the cell wall of bacteria or within those bacteria in form of their nucleic acids. Or perhaps in some cases, these are also present in viruses, as an example, not necessarily LPS, but CpG could actually be present in viruses. You can, in fact, reduce virus load and virus shedding. So this could actually be something that can be used in an outbreak situation, as an example, I think, possibly. One can also utilize the same sort of molecules in addition to vaccines. And in fact, in human trials, some of these molecules have actually been proven to be quite efficacious in terms of inducing responses to vaccines, various different types of vaccines. So we are currently working, and I don't know if Shireen is here. So Shireen Singh, oh yeah, here she is. Shireen Singh has also been working on development of some of these novel vaccine formulations that incorporate some of these adjuvants. One of the things that is of interest is how to get these things inside the chicken. Because obviously nobody is going to be picking up, let's say, one chicken at a time in a 500,000 broiler farm and inject them with any of these ligands. So one can envisage the use of, for example, the in ovo route, or perhaps trying to get these things through a spray route or a respiratory route, or perhaps through feed, water, and so forth to chickens. And Tamiro Alki has been working on packaging some of these things, some of these ligands, in order for better delivery of these compounds into the respiratory system or to the intestinal tract. I'm just going to pass through this very quickly. We are also working on development of probiotics, and we've identified commensal bacteria that have immune-enhancing abilities, and they can have the ability to reduce by a few orders of magnitude burden of pathogens. And I think, Jen, you can correct me if I'm wrong, but last time I checked, I think we can actually reduce burden of salmonella by almost 1 million times when we administer probiotics prior to the time of administration of salmonella. And in fact, this particular formulation is under investigation by an industry partner at the moment in the UK. The other aspect of the work that we are currently working on is aspects of gut health is to develop methodologies for reducing Campylobacter burden. I became interested in Campylobacter because obviously Campylobacter is one of the major causes of foodborne illness in humans. The last statistic I found was in 2004, and there might have, and I'm sure that there would have been some additional statistic, but this is the last one I found. And based on this, in Canada, we have approximately 10,000 cases of campylobacteriosis that are reported, and there are probably a few others that don't get reported. So I think Campylobacter is a problem. Chickens do harbor Campylobacter, and as many of you know, Campylobacter in chickens doesn't cause any disease. So as a result of that, many of them are carriers of Campylobacter, and through the processing of carcasses, Campylobacter can adhere to poultry products, and as a result of that can be passed on to consumers. And this is a cause of concern for poultry producers and poultry industry as a whole. And we are currently working on developing various different formulations for potential vaccines against Campylobacter. And where is Khaled? Here we go. Here he is. And Khaled is currently working on developing some of these formulations with the help of Doc Hodgins. I also wanted to touch on some interesting topic that we've recently started working on, which has to do with avian influenza and the interactions between avian influenza and commensal bacteria. I didn't tell you, but commensal bacteria do have a lot of interactions with the host immune system, and they can upregulate or downregulate the host immune system. And a student in my lab, a PhD student in my lab, Alex, has recently joined the lab, and he's been working on the connection between commensal bacteria in the gut and how the immune system is triggered in the gut in response to avian influenza. But that's still in rudimentary stages of development, and we've forged a collaboration with Sick Kids Hospital with Dr. John Parkinson, who's going to be working with us on this particular subject. So we are going to be doing some microbiome analysis in the future. And last but not least, I've got to tell you that I didn't do any of the work that was described in my presentation. I did absolutely zero. I haven't even touched a pipette for a long time, and I have no fear in terms of accepting the fact that I've not really contributed to that research. I've been the man behind the closed doors sometimes running the research lab, but this is actually my research lab at the moment. Shirin, Bahram, Ravi, who you know, Doug, Khaled, Mohammed, who was our undergrad student during summertime, Vanderlei, Alex, Tamiro, Ruby, Neda, Adriana, and Ayumi, and that little guy who was just visiting us. So that's all I had to say, and my apologies if I went a little bit over time. Thank you.
Thank you very much, Shayan, for talking about the network as well as your research program. I also take this opportunity to thank many of you who contributed to provide us with the fact sheets and the research stories that we're publishing in our on our website, as well as sharing the stories with PIC and everything. So now we can open up for discussions, and feel free to ask questions.
And perhaps, before we start, that part, I was also just going to say that, the main purpose of today is really, to, I would say, intermingle and, talk to each other. But this is only a forum, to open channels of communication. But if you do have some questions for me, I'm more than happy to answer. Yes, Suresh.
This is more of a non-technical, futuristic question. I know golf plays a significant role. We have AHL, which contributes significantly to the poultry industry. In terms of taking the PHRN to the next level, how do you envision? Are we looking at becoming a bigger entity such as an NCE, National Center for Excellence for Poultry Health Research, or even to a much bigger global level research collaborative platform?
You're actually putting me on spot here. Yes, absolutely. So there are two things here, Suresh. If you're asking what my hope is an NCE. Would we actually be able to get an NCE? I think, if One Health NCE, which was actually spearheaded by University of Saskatchewan, and we had a major contribution to that, was not successful, I think, probably a poultry health research NCE, it's unlikely to be successful. But that's my hope, and I'm not going to give up my hope. But I think, we also have to be realistic about it. So, and I think at the same time, we should not discount, other opportunities. Since you asked me about NCE, I can't actually, envision, an NCE at the moment unless, we see some good signs. If you're talking about an NSERC strategic network, I think we are actually in a very good position, perhaps, for next year to apply for an NSERC strategic. And within the umbrella of NSERC strategic network, then we can fund projects. I think that's definitely a possibility, because we do actually have some very good examples of that. Like, for example, the mastitis network. Like, for example, bacterial diseases network of pigs. And I haven't actually been, I haven't been involved in any of those, but I know that at least, there have been some good, successful examples. So if I were to answer your question, Suresh, I would actually say we should actually, steer in that direction, in the direction of an NSERC strategic network, and we have to justify why this network is needed, is timely, and we can actually lead that network. And like I said, if we can make that justification, I think we can get in the neighborhood of $1 million a year for running the strategic network. But I think, there are also other opportunities, funding opportunities to tap into. And I think, we need to tap into those. Do I see PHRN as a funding agency? I guess, that would be also an interesting proposition. It wasn't actually meant to be a funding organization. It wasn't actually meant to fund projects per se. But I think, this is actually something that we should definitely get together at some point of time and talk about aspirations for a PHRN and what can actually come out of it. So can we actually make this global? We are actually making it global. So just to give you an idea, there is actually a global movement now on the European side, not so much on the North American side or South American side, to put together networks of people within Europe and parts of Asia and parts of Africa. I am actually in contact with the person who's leading that. And I think we can actually, in fact, become the North American hub for that initiative. We have also been involved in conversations with the CRC poultry, with all its I guess negative connotations associated with the CRC poultry in Australia. We have actually been in contact with them. They're also going through their own transformation phase because they've just ended a funding cycle. So I believe they're also doing a little bit of soul searching to see what exactly they want to do in the future. And I think we can and we should be connected to CRC poultry in Australia. We should be connected to the European initiative, the GROD initiative, the Global Avian Research Initiative in Europe. And obviously, in my opinion, we should actually be the hub for a global initiative within North America. That's just my hope. But I think we do actually need quite a bit of outreach at the moment. And in order to do that, we need to have funding in order to reach out to all various different organizations. Yes, Eva.
Yeah, you mentioned, Shayan, thank you very much. At least you set the tone, how to run this, or what kind of talks to give. So that's really good. On that line, so Ravi, maybe you can send to the speakers actually the template for the slides.
Absolutely.
So that would be a uniform slides or the way people would use when this presentation is put on the website. So that would be good kind of course the same thing or from the logos around there.
Absolutely.
We were talking about the official recognition of the network over here, so by the university. We mentioned that the Office of Research is supporting. So when is this official recognition going to be? So when we can all send ourselves center, not network, so we can use that logo or on our stationery, not necessarily that it's going to be part of the network, because I think that would be a really huge visibility.
Okay, so I can actually tell you, maybe I should go off record here, Kevin. No, I'm actually joking. I can be on record. My first priority, as you know, our new dean just arrived a couple of days ago, actually last Thursday, I believe. My first item on the agenda is actually to book an appointment with the dean and just go directly to the dean and ask for some sort of official recognition, because the idea of having a center or an institute is a good one, and it has actually been discussed with our administration. I think there was a bit of a resistance in the past, partly because of, I think, the history of some of those institutes or centers that have emerged, and because of lack of funding, they've kind of actually languished, in somewhere in the no man's land. So as a result of that, there was a bit of a, I would say, hesitation to identify this network as a center or an institute, because there was, the thought that there may be, some obligation on the part of the university or the college. But the ironic part of this is that there is already a commitment. Like I said, the college has already committed funding, or the department has actually already committed funding to this project, to this network. So it's not as if, there is no commitment, financial commitment. So that's why I think first order of business for me is to actually go to the dean and perhaps, just make a proposition to the dean that we need to have more formal recognition. The second thing that has actually been put in the back burner for some period of time, and in some of my recent conversations with Tim Nelson, we concluded that it would be a very good idea to restrike the advisory board and get the advisory board engaged quite intimately in the process, so that we can not only get opinions of our community at large, but we can also get more refined and polished advice from an advisory board. And we do actually have an advisory board, but the advisory board needs to be re-engaged, and their advice needs to be sought. Jim?
Yeah, I know this is the right time, but I just wanted to make a bit of an announcement. So as chair of the new Department of Animal Biosciences, so we're not animal and poultry science anymore, we're animal biosciences, which we're pretty excited about. That doesn't mean we don't care about poultry. We have a lot of poultry people, and I also want to announce that we are now successful in appointing somebody to the poultry efficient position that we've been actually gone 3 rounds for, and you'll be hearing more details about that in the near future.
On that note, Jim, actually, that we just got the pathologist new newsletter, and it's on the front page that your department changed its name. So that's to bring this.
Been a long time coming. It really reflects the sort of breadth of things that we do now. We're not changing what we've done. We're just acknowledging where we've gone.
And by the way, Jim, obviously English is my second language, but I always thought that poultry are actually animals. Are they not? Last time I checked, I think they were actually. But I guess, there was some sort of a distinction, that was made probably, 70 years ago or something like that. I think I read in one of your comments, which was on the website, on the main website, that, this name actually came about maybe 60, 70 years ago or something like that.
I think in the 68 or 70, when the poultry, husbandry, and animal departments came together, they couldn't agree on a name, so they called it Animal and Poultry Science. So that's where it came from. So now we finally named the department appropriately.
Animal Biosciences.
Animal Biosciences, yes.
Excellent. Yes, John.
Very, very nice talk, Shayan.
Thank you.
In terms of the advisory board, I think it's a very good idea. We get funding from the Canadian Poultry Research Council, which has matching funding from AAFC, Agriculture Canada, under the Growing Food. And I know they have a lot of money, or they had a lot of money. I guess we spent most of it for them. But I think on an advisory board, it would be really useful to have CPRC involved, because I think they help to have a national picture. Like we have international connections, but actually there's a lot of really good work going on across the country, which some of us may know about, some of us don't. So I think CPRC are very important.
Absolutely, and I totally agree with you, John. And as a matter of fact, we have actually tried to reach out to some of our colleagues in various different provinces of Canada, and currently we do have colleagues from, for example, Saskatchewan, from Alberta, from PEI, and so forth. We should have representation or complete commitment, by all these various different veterinary colleges. We have 5 of them, and I believe last time I checked, I think we have 12 or 13 agricultural colleges. I think, ideally we should have commitment and representation on our roster, from all these various different provinces, and as well as, having them on board within the advisory board. Yes, Eva.
Yeah, along the same the productions that we're supporting, maybe we shouldn't forget about OMAF.
Yes.
That would be very important, and I guess as well as funding and actually contribution towards our stations as well too.
Yes, absolutely. One of the things that we thought in the very beginning that it would be a very good idea to do, and I think we still need to strengthen that, was to really put things like bits and pieces of the puzzle. So in order to have a very active research program, a globally recognized, internationally recognized research program, what do we need to have? We need to have good people, we need to have good facilities, and we need to have good ways of communicating our results with our colleagues and peers. And one aspect of it obviously was facilities. And I've tried as much as I could, and I think when Ravi also came along, he and I have had the same conversation, that we need to catalog what we have, because when you look at the amount of investment that either the province of Ontario through OMAFRA or through ARIO or otherwise have done for University of Guelph is absolutely enormous. I don't even think that any other university would even come close to us. And in fact, we've been actually working with OMAFRA to create a list of spreadsheet of all the various different facilities and where exactly we are with respect to our capacities and capabilities. So I totally agree with you, and I think this is not just for facilities for housing and maintenance of our animals. This also goes beyond that. Every research lab. What do we have in each research lab, and why our research is better than our competitors, let's say south of the border or elsewhere in Europe? And I think it really boggles my mind how much investment we've had in poultry research as a whole, both in terms of facilities, infrastructure in terms of isolation, RKL, and so on, and also within each research lab. And Eva, you also brought up another interesting point that I think poultry has to be involved very intimately and very strongly. And that actually has to do with the future of two of our facilities, two of our infrastructures, RKL and the isolation unit. And what exactly is going to happen to those? I think the PHRN and poultry researchers as a whole, we have to be involved. So I think I've said too much now. And like I said, the whole purpose really was to intermingle and say hello to one another and just to know who's actually...
Enjoy the coffee.
And enjoy the coffee. Yes, of course, enjoy the coffee.